In this lesson, you'll learn about genetics and the basics of monohybrid and dihybrid crosses. Okay, genetics is how traits are passed down from parent to offspring. It's how traits are inherited from their parents through their genes. But what exactly do we use in terminology to express what people have in their genes? Well, that word is called a genotype. A genotype is a combination of alleles that you have in your DNA. You inherited one allele for a certain gene from your father and one allele for a certain gene from your mother. The genotype are those two alleles, each of which were inherited from your parents. And the genotype is held in your genes within your DNA. What's an example of an allele? For example, eye color gene. There are several different alleles for the eye color gene, including brown, blue, green, hazel. Those are all the different colors that you can have of eyes. Another gene, hair color gene, examples of alleles for hair color gene would be brown, black, red, or blonde. Those would all be alleles for the hair color gene. If you have two of the same alleles for a particular gene, then you're said to be homozygous. You can either be homozygous dominant or you could be homozygous recessive. In the case of eye color, I'm going to use the letter E to represent the eye color gene. So two dominant alleles, two big E's, would be what you inherited from your parents. So your parents would have both passed you a dominant E allele for eye color. In the case of the recessive alleles, if you inherit two recessive alleles from your parents, you're said to be homozygous recessive. That's your genotype. Two little e's is a homozygous recessive genotype. If you inherit two different alleles from your parents, one dominant allele and one recessive allele, this is said to be heterozygous. One big E, one little e. You'll still show the dominant color, but you will be a carrier for the recessive trait, which is the little e. The other word that we use in genetics to describe the traits of a person is called a phenotype. A phenotype is the physical traits of an organism. I always remember the difference between genotype and phenotype because genotype is found in your genes. It's the actual alleles that you inherited in your DNA. Phenotype, on the other hand, is your physical traits, what you actually look like. So some examples of phenotypes would be taster versus non-taster. So when you guys did the PTC testing paper, or for those of you who are absent, we tasted a chemical on a piece of paper. It's genetic whether or not you can taste it. Some people are tasters for PTC, so they got a very bitter taste from the paper in their mouth. Other people are non-tasters. They can't taste the PTC on the paper. I am a non-taster. That's my phenotype. I cannot taste that bitter chemical. Another example of phenotype, dealing with Gregor Mendel's genetics. Gregor Mendel was an Austrian monk, and he worked with pea plants in a monastery back in the, I think, 1500s or 1600s. He's coined to be the father of genetics because he followed the traits of pea plants from parent to offspring. And one of the genes that he worked with was flower color gene. And he found that there's two phenotypes for flower color gene, purple or white. Purple was the dominant phenotype, so if you inherited or if the plant inherited at least one dominant allele for purple flowers, the plant had purple flowers. But if the plant inherited two recessive alleles that code for white flowers, then the plants would be white. Like I was just saying, some alleles are dominant over the others. Recessive alleles have their expression of their trait coded for by the allele that is masked by the dominant allele. So recessive alleles are covered up by a dominant allele. Dominant alleles, on the other hand, are the alleles that cover up the expression of the recessive allele. So if you inherit a dominant allele, it covers up your expression of your recessive. So in the case of eye color, brown is dominant over blue. So if you inherit a brown allele from one of your parents, then your eyes are going to be brown. Even if you inherit a blue allele from one of your parents, your eyes will be brown because brown is the dominant color. And like I said, in the case of pea plant colors, purple is dominant to white. So purple is the color of the pea plant if they receive the dominant purple allele. So how do we predict the traits of offspring? Now we know genotypes are found in your genes. It's the alleles that give you certain traits. Phenotypes are what an organism looks like. How can we figure out how traits are passed down from parent to offspring? Well, we use a tool called a Punnett square. Punnett squares show the outcome, the possible outcome of a genetic cross. A genetic cross is simply the mating of two organisms. On the screen here is an example of a Punnett square showing how a homozygous recessive green pea pod was mated with a heterozygous yellow pea pod. Yellow is the dominant color of the pea pod, which is expressed by that big Y allele, and green is the recessive color. How do we get to this point, though, of the Punnett square? So when you set up a Punnett square, you're just going to make a box that's got 4 squares in it, just like this. And in this case, our first parent was homozygous recessive, so that's why they use 2 little y's. So every person or every organism has 2 alleles for a particular trait. In this case, it's 2 little y's. The second parent that was being mated in this cross was heterozygous, so that parent had 1 big Y, 1 little y. Now, when you cross these in the square, you bring down this allele in this direction, and because it's recessive, I'm going to write it second. And again, you do the same thing over here. So you carry those down, and it's like that. Now this allele right here, you're going to carry it across, so now I write a big Y here, a dominant Y here, and same thing down here, you just carry those across. Little y, little y. So what the Punnett square shows us is just the probability of getting certain outcomes from a genetic cross. It does not tell us how many organisms or what they look like, it just tells us the probability. So in this case, if you cross a homozygous recessive green pod plant with a heterozygous yellow pod plant, you have a 50% chance of getting yellow pods because that heterozygous genotype that's right here, these are going to give us yellow pea pods. So this is 50% yellow pea pods. Then that means that we would have a 50% chance of getting green pea pods because the only other option of color of pea pods in this case is right here. Both of these genotypes give us green pea pods, so we're 50% green. Okay, so again, a Punnett square is just a tool that geneticists can use to predict the possibility of getting certain outcomes from a genetic cross. When we do single gene crosses, so crosses between organisms and we're only looking at one gene, this is an example of Mendelian genetics because when he did his crosses, he crossed pea plants, but he only looked at one gene at a time. So some of the genes that he looked at were flower color, flower position, seed color, seed shape, pod color, and stem length. And he found that these traits were all passed on with simple dominant and simple recessive alleles. So in the case of flower color, purple is dominant over white. For the flower position, axial flowers, which are on the side of the flowers coming from the side of the plant, that's dominant over terminal flowers, which are at the top. In the case of seed color, yellow is dominant over green. Seed shape, round is dominant over wrinkled, and so on and so forth. Now, you can see here that there's a parental cross, and that's in the top. This was 2 parents. This is the first parent. That's the second parent being crossed. And these are the offspring of those parents. So what we call the offspring of the parental generation is known as the F1 generation. Now, if we were to cross 2 organisms from the F1 generation, then that would give us the F2 generation, and so on and so forth. F stands for filial, which means son in Latin. So, yeah. Now, when he crossed these pea plants, they were all homozygous for their traits, the parental generation. So this would have been a homozygous dominant purple plant crossed with a homozygous recessive white purple plant. And all the F1 generation came out purple. That means that these F1 generations had one dominant allele, one recessive allele, and we call that a monohybrid, So if we would have crossed two F1 generations, that would have been a monohybrid cross, which is a cross between, excuse me, which is a cross between organisms with two different alleles of a trait controlled by one gene. Now is a good time to pause the video and work on some monohybrid crosses. When you're ready, you can come back and work on dihybrid crosses. Dihybrid crosses are crosses that are going to be between organisms, and we are looking at two genes this time instead of just one. Technically, a dihybrid is an organism that is heterozygous for two traits controlled by two genes, but sometimes the phrase dihybrid cross is loosely used to talk about a cross that's between two organisms with two genes, looking at two genes. So here's an example of a dihybrid cross, crossing two organisms that are both heterozygous for seed shape, round versus wrinkled, and yellow seeds. So the first organism here is big R, big Y, little y. The second organism is the same thing. This is a dihybrid cross. You're crossing two dihybrid organisms. So the first gene is right here. This is the gene for seed shape, round or wrinkled. The second gene is right here. This is the gene for seed color, yellow versus green. Now, in order to set up the Punnett square, you have to use a method called the FOIL method to determine the possible gametes that will be crossed. So remember, in meiosis, the gametes are going to be made up of one allele for each trait because each parent only passes down one allele. So that means that each of these parents here are only going to pass down one allele for seed shape and one allele for seed color. So let's look at how to use the FOIL method. Here's my genotype of the parents. And FOIL stands for first, outer, inner, last. So the first possible gamete that this parent can pass on is the first two alleles of each gene. So this parent can pass on a big R and a big Y. Or if we look at the outer two alleles, they could pass on a big R and a little y. Or if we look at the two inner alleles, the parent could pass on a little r and a big Y. And if the parent passes on the last alleles, then they're going to pass on the little r and little y. So how does this get set up in a Punnett square? You have to draw a larger Punnett square for this because you're crossing two genes. So like I showed you before, using the FOIL method, we determined the possible gametes that each parent could pass on. That's what these are up here. These are the possible gametes that the parent could pass on. Now you have to combine them in the Punnett square. So you do the same thing as you did before. You bring down the first letter, you bring across the second letter, and so on and so forth. And you do that for every single square. Now when you do this, it doesn't really make sense to look at the genotypes alone. It makes sense to look at the phenotypes alone. So when this particular person did this Punnett square, they looked at round and yellow seeds first. And they wanted to see how many out of 16 give us round, how many genotypes out of 16 give us round and yellow seeds. So there's one here, there's one here, 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 and right there. That's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 out of 16 offspring are predicted to be round and yellow. If you looked at round versus green, here's that. Round versus green's right here, and right there. So 3 out of 16 would give us round versus green. Wrinkled and yellow is right here. So there's 1, 2, 3. And then only 1 out of 16 would be predicted to show both recessive traits. So wrinkled and green seeds. So every time actually you cross two dihybrids, so two organisms that are heterozygous for both genes, you'll always get this 9:3 to 3:1 phenotypic ratio. So that may be helpful when you're doing problems on a quiz or a test.